kids, good evening and welcome back to Yupp Master, and these are the foundation courses. Now, if you remember, we've started with the whole unit of cell, and today is in fact our very last lecture for this unit of cell, its structure and functions. And here in our last lecture we described all about, we took a revision about the prokaryotic cell, and we also went ahead and discussed some MCQs. So let us do the same for our eukaryotic cell. We first go ahead and we describe, or we'll go through a quick look at all the different structures which are present in the eukaryotic cell, and then we'll go ahead and discuss some more MCQs. Okay, so are you with me, and shall we start? All right. Now in the eukaryotic cell, the outermost membrane, okay, in an animal cell is that what is called as a plasma membrane or a cell membrane. All right. Remember, when we talk about plant cells, over there the outermost covering is going to be the cell wall. But both animal as well as plant cells are having plasma membranes. All right. Now, the point which we need to remember about plasma membranes, which can come as MCQs, let's see what those points are. First of all, the scientist names. Okay, the names of the scientists here, Singer and Nicholson. These are important scientist names which oftenly come as the MCQs. All right. Also, plasma membrane is called as a fluid mosaic model. All right. Plasma membrane, fluid mosaic model. Now, do you remember what that plasma membrane was made up of? It was made up of a phospholipid bilayer, and there were proteins too. All right. So let's see here. Here's the phospholipid bilayer and the proteins. So where are those over here? This is the bilayer, the phospholipid bilayer, and this over here is the protein. All right. Now we see here that when we're talking about a phospholipid, okay, only the phospholipid, then it has got a head and it has got two tails. Now the head part is the phosphate part, the tail part is the lipid part, and the head part is the part which is going to allow water to come in the cell. So that is called as, it is a hydrophilic head, but the tail regions are going to repel water. So you can call the tails as two hydrophobic tails. So the hydrophilic head, the hydrophobic tail together are forming one phospholipid, and put them all together forms one layer. The same way, there is one more layer. That's why we're going to be calling it as a phospholipid bilayer. Okay, it's a bilayer. All right. Then we see that the proteins that we mentioned which are present on this plasma membrane can be of three types. Which are those three proteins? The extrinsic ones, which are found externally. Intrinsic, which are going to be completely embedded inside that phospholipid bilayer. And then there's going to be a protein which actually goes through the length of that whole plasma membrane. And that protein is going to be called as tunnel protein, or you may also call it as the channel protein. Why? This is the protein through which any substance can either enter into or go out of the cell membrane. Alright. So those three proteins we've studied about. Then more importantly are the functions. In the MCQs, most of the MCQs are oriented directing towards their functions, which is the cell organelle and what is their function. Okay, so when we talk about the cell membrane or the plasma membrane, what exactly is the function of the plasma membrane? First, it is going to be separating that protoplasm. What is the protoplasm? Do you remember that? Here was the cell, here is the nucleus, all right. This here is the nucleoplasm. This over here is the cytoplasm. Together here, this is going to be collectively called as the protoplasm. So the cell membrane, this membrane over here, the outermost membrane, that is actually going to be creating a barrier between the external environment and the internal protoplasm. So it is doing the work of separating it. Then it is the reason that there is a particular shape to the cell, and it also provides protection. Okay, it is semi-permeable, hence it allows certain substances to come in, doesn't allow certain substances to come in. And how are the things coming in or going out? Remember that particular type of protein. Which protein was that called as? It was called as the tunnel protein, or we called it as the channel protein. Alright. Now, Also, we see that there are many membranous proteins which are present, which also serve as enzymes. Now, if they are working as enzymes, then this means that they're going to be enhancing any metabolic activities which need to be taken at that point. Okay. Then, in unicellular organisms like the amoeba, the cell membrane is actually going to form the pseudopodia. So, when we talk about amoeba and when they want to take food in, then how are they going to take food in? Well, the cell membrane is going to just change its shape and in such a way that it is able to engulf that food substance. So, it is taking it in, alright. So, this process where it is going to be taking food in can be called as endocytosis, and when it wants to throw food out, you may call it as exocytosis, or rather wastes out, alright. So, this whole process of taking food in or throwing waste out is going to be done by the pseudopodia found in amoeba, okay. Now, moving on towards the cell wall. Cell wall, remember, it's present only in plants and in fungus, okay. It is absolutely absent in the animal cells. The cell wall is the main reason why these plant cells actually have their shape, okay. Now, what is it that we needed to remember about the cell wall? Remember, this in plant cells, this is a layer which is outer to the cell membrane or the plasma membrane, okay. So, the outermost layer of a plant cell is going to be the cell wall. Inner to the cell wall is the plasma membrane, okay. Now, the cell wall between two adjacent cells have got these bridges in between them, okay. In order for any substance to be transported from one cell to another cell, there has to be some sort of bridge in between. Now, it is these cell walls at the level of the cell wall where those bridges are present, here and here. So, that part there, the bridge over there, is going to be called as plasmodesmata. What is it called as? It's called as plasmodesmata, which is going to be allowing to transport substances from one cell to another cell, okay. That is plasmodesmata. Alright. Next, moving on towards the cytoplasm. Do you remember that the cytoplasm was a semi-solid jelly-like substance, okay, and it was a colloid substance, which means that when you observe it under the microscope, it looks like it is just one big substance, one big jelly. But inside that jelly, there are various cell organelles embedded inside. But the cell organelles are not dissolved. They are in their own state. That's why we're going to be calling it as a colloid-like substance. Okay. Then we see that it shows streaming movements. The cytoplasm of a eukaryotic cell is having the ability to circulate within its cell, okay. What is it going to be doing by doing this? That any nutrient, if it is in one part of the cell, by the movement of cyclosis, by the cytoplasmic streaming movements, then that substance, that nutrition can be delivered to all the different parts of the cell. So, this is an important factor which is present only in the eukaryotes. Remember that in prokaryotic cells, cyclosis is absolutely not happening, okay. Now, next cell membrane or the next cell organelle, very important cell organelle. That's why I'm going to go ahead and will revise all the points of this mitochondria. Well, mitochondria is the basic powerhouse of the cell, the place where we get all of the energy. What energy? Energy in the form of ATP. That is the basic powerhouse of the cell. This is the region where ATP is made. Now, the mitochondria, how many are there? Around 50 to 55,000 per cell. Now, one thing I want you to remember here, that there were two membranes. There is an outer membrane and there was an inner one. Remember that the outer membrane is going to be freely permeable, but that inner membrane is the one which is semi-permeable, okay. So, outer membrane is freely permeable, inner membrane is semi-permeable. So, the inner membrane is actually going to be controlling what enters in and what leaves the cell. Inside that inner membrane, you can see the presence of all of this here, and that is going to be called as the mitochondrial matrix, alright. So, that mitochondrial matrix is there filled inside the inner membrane. Now, also one thing, the inner membrane is thrown into folds, okay. Do you remember the names of those folds? They were called as cristae. Okay, now these folds are actually going to be having certain substances on it. What attached to it? Those are called as small substances called as the oxysomes. Either you call it as the oxysomes, or you may also call it as the F1 particles, or we may also call it as the elementary particles. Oxysomes, F1 particle, or elementary particles. Elementary. Let's see how that oxysome looks like. This what you see here is an oxysome. Can you see how all those oxysomes are placed on that inner cristae membrane? Okay, so now this might make it clear why exactly the inner membrane of the mitochondria is thrown into folds. So the more the surface area is, the more amount of these F1 particles can be accommodated. So in this diagram over here, this what you're seeing is the cristae, and these over here on top are the oxysomes. The oxysome is the site where actually ATP is going to be synthesized, alright. So the more number of oxysomes present in that mitochondria, the more amount of ATP it is going to be able to make, and that is why we have an inner membrane which is of infolds, alright. The next cell organelle we're going to head on towards to is the endoplasmic reticulum. This is also found in all the eukaryotic cells, not in prokaryotic cells at all. They are not also found in the human ovum and in the mature RBCs. Now, remember one thing, this endoplasmic reticulum starts from one place and ends at another place. Where is it going to start from? It always starts at the level of the nucleus. That nucleus is going to give rise to the endoplasmic reticulum always. And remember one thing, there is something which is called as an endomembrane system. An endomembrane system. What is this all about? It means that it is the structures or the list of structures which are going to start at the level of the nuclear membrane. After nuclear membrane comes the endoplasmic reticulum. After the endoplasmic reticulum will come the Golgi bodies. Those give rise to the small vesicles, okay, and that is going to give rise to the lysosomes, and that is going to ahead and it would go and connect with the cell membrane. So basically this is a whole chain of structures which are arising one after the other. Those are going to be called as the endomembrane system. Endoplasmic reticulum is going to be forming from the nuclear membrane. Now, what is the function of endoplasmic reticulum? It is the site where glycogen and lipids, glycogen is a complex form of glucose, okay, and lipids, these are going to be synthesized here, and they are also going to be stored at the level of the endoplasmic reticulum. Now, next, after endoplasmic reticulum comes the Golgi complex. This Golgi complex we see, these are concerned with the secretion of glycoproteins. Remember the function of Golgi bodies. They're going to be secreting glycoproteins and also glycolipids, and they're going to be helping in the storage of carbohydrates, proteins and fats, alright. So, secretion of glycoprotein and glycolipids and storage of carbohydrate, proteins and of lipids, okay. Also we see that they are the ones which are going to be taking all those simple molecules, putting them together and forming the complex carbohydrates, okay. Now, after endoplasmic reticulum, we found that it gives rise to the Golgi bodies. The Golgi bodies are going to be giving rise to smaller ones. Those are called as the lysosomes. What are lysosomes? These are actually suicidal bags, okay. Now, ever heard of an army officer who is quite aged? Do you think that he, after all these years of service, do you think that he would actually like someone to come and tell him that, no, now you sit at home, you're too old? No. So what does he do? Doesn't he go forward and he himself is going to take a voluntary retirement? So basically, this is exactly what the lysosome is going to do in that cell. That the lysosome is going to, if at all there is something wrong with the cell, or if the cell has to destroy itself, or even if you see that the cell has got maybe too many cell organelles which are not required, then the lysosomes will be taking a role play here. What are they gonna do? They are containing an enzyme which is called as acid hydrolase. This acid hydrolase is an enzyme which helps to break down any material present inside the cell. So basically this lysosome, if at all its membrane is broken, is opened, then the acid hydrolase enzyme would be released and that would cause the cell to just degenerate. And this is the reason why the lysosomes are called as suicidal bags. Where do these lysosomes actually originate from? They are the ones which are originating from the Golgi bodies. Golgi bodies came from endoplasmic reticulum. Okay. Now next, the next cell organelle we studied was called as the cytoskeleton. Okay, cytoskeleton. This is the basic framework of the cell. Remember that the cytoskeleton had three parts to it. First part was called as the microfilament. Second part was called as the microtubules, and the third part was called as the intermediate filaments. Okay, they three were made up of their own respective proteins, actin, then tubulin, and then keratin. So these were basic proteinaceous structures. Moving next to the cell organelle, we call this as the ribosome. And remember, ribosome is a cell organelle which has no membrane around it. So it is not a membrane bound cell organelle. Hence, I will say that this can be found even in the prokaryotic cells. Okay. Now, so this is one thing you need to remember. Ribosomes are found in prokaryotic as well as in the eukaryotic cells too. Okay. Now, we see that at different locations and even at different cells, the ribosomes can be different. First, when we talk about in the eukaryotic cells. Okay, in eukaryotes, the whole ribosome, where I am talking about both the large and the small subunits together, they are going to be settling at a rate of 80S. So here it is 80S. The large subunit settles at 60S in eukaryotic cell and the smaller subunit settles at 40S. Talking about in a prokaryotic cell, that very same ribosome on a whole settles at 70S. Then the larger subunit settles at 50S and then the next one at 30S. Mitochondria, the whole 55S, 39S and 28S. And then plastids, 70S, 50S as the large subunit and 30S as the smaller subunit. These, the valuations of the ribosomes, their settling rates as a whole, the large subunit, the small subunit is something you're going to have to remember for MCQs. Any of these can be in the form of an MCQ, the whole equation. So remember these four, alright. Okay, now after ribosomes, we move on to the next cell organelle, which is very important process or a very important cell organelle with respect to cell division. Okay, with respect to cell division, this is an important cell organelle. What do we see here? Over here, this is a cell with cytoplasm. And can you see how these centrioles are located here and they are actually placed perpendicular to each other? Can you see that? Yes. So we see that these also are non-membranous cylindrical structures. They are found in the animal cells and they help in spindle formation. When during cell division, they are going to be helping in spindle formation. Now, these cell organelles are called as centrioles. But do you see this over here? This is a name called as centrosome. What is that centrosome? Well, the centrioles are located in the cytoplasm, which is very near to the nucleus. So the centriole inside that cytoplasm of that area is going to be called as centrosome, and the centriole inside the centrosome together, okay, or rather the centriole along with this cytoplasm over here, that is called as the centrosphere. Together, those are going to be collectively called as the centrosome. So centrosphere is the cytoplasm, inside that is the centriole, together that whole structure is going to be called as the centrosome. What is centriole useful in? In the spindle fibers formation. Okay, there are these spindle fibers which are formed during cell division, that is formed by these centrioles. And also we see that the centrioles, the arrangement, remember these are structures which are completely non-membranous. Okay, so they have actually a cluster of 9 tubules. So here these are 3 together. This is one triplet, another triplet, another triplet. Similarly, there are going to be 9 triplets over here, okay? And all of these 9 triplets that you're seeing here are arranged in a circular pattern. So, can you see that these triplets, what I'm saying exactly, these are made up of microtubules. What are these? Proteins, okay? These microtubules are responsible for making up this whole centriole. Triplets, and 9 of them, all of them are placed perpendicular to each other, and hence it is going to be forming a circle. Is there anything present in the middle of the circle? Absolutely not. There is nothing present in the middle of the circle. That's why we call this whole arrangement as a 9 plus 0 arrangement over here, okay? That is what present in the centrioles, alright. Now, we also saw that there is another cell organelle which is called as the vacuole, and that vacuole is present only in, or rather it is present in a large, abundant, prominent way in only the plant cells, okay. It is present in the animal cell, but in animal cells it is comparatively smaller and it is temporary, okay. Now we see here that this has a single membrane, okay. It is not a double membrane, just single, and it is filled with fluid inside. Now that membrane that is forming it, that membrane is called as the tonoplast. The fluid inside was called as the cell sap, okay. So in animal cells, these are small and temporary, whereas in the plant cells, they are large and permanent, okay. They are bound by tonoplast. They are filled inside with the cell sap. And what does this cell sap contain? It contains good things and bad things too. What are the good things? Salt, sugar, amino acid, proteins, pigments, and also it contains waste material, alright. That was all about the vacuoles. Now, we also went ahead and we learned in detail about the nucleus. Let's just have a quick recap of what were the parts present of the nucleus. First and foremost, the double layered nuclear envelope. Remember, there were 2 layers, okay. Both the layers, the outer membrane and the inner membrane, were separated by a middle space. That space was called as the perinuclear space. Next, we saw that inside the whole thing it is filled with a cytoplasm, which we're going to be calling as the nucleoplasm. Then there is another organelle inside here which has actually no membrane around it, and that is going to be called as the nucleolus. And the last, these small threads that you're seeing all around here, those threads are called as the chromatin network. These are the network, or these are those small threads which are going to actually condense together, and that too it's going to be forming a chromosome, but only when the cell wants to divide. When the cell is not dividing, we see that the chromatin network is found just like this, okay. So we've gone ahead, we've learned and we've discussed all the important functions and the structures of the various cell organelles of eukaryotic cells, okay. Now it's time, if you're all with me, it is now time that we go ahead and we can discuss certain important MCQs from the eukaryotic cells. So are you all ready with me? Okay, let's see the first one. Which from here is a part of the endomembrane system of eukaryotic cells? Remember, endomembrane system, starting from the nuclear membrane, going to the endoplasmic reticulum, to the Golgi bodies, and then to the forming the lysosomes. So in these here, what is the correct answer? Is it mitochondria, perioxysomes, chloroplasts, or Golgi bodies? Endomembrane system would be the Golgi bodies. So correct answer here will be Golgi bodies. What other structures are present in the endomembrane system? They would be the nuclear membrane. Then you have the endoplasmic reticulum. You have the Golgi bodies. And finally the lysosomes. Alright. Okay. Next, rough endoplasmic reticulum is rough because it contains, remember there were 2, there was a smooth endoplasmic reticulum and there was a rough endoplasmic reticulum, okay. Now, the rough is called as rough because it contains ribosomes, okay. So, due to the presence of those granular ribosomes on them, it has a rough appearance. Maximum it was found in the liver, okay. So over here it is rough because it contains the ribosomes. Okay. Next, which cell organelle connects the nuclear membrane with the cell membrane? So over here if you have the cell and this here is the nucleus, arising from the nucleus connecting all the way to the cell membrane, there is one cell organelle, and that cell organelle which I am drawing right here, it is actually forming like a big framework around the cell, inside the cell, and that can be called as, tell me what am I drawing, tell me what am I drawing, isn't this a structure which is called as the endoplasmic reticulum? Okay, so it is this endoplasmic reticulum which is able to connect the nuclear membrane inside all the way up till the cell membrane or the plasma membrane, which is the outermost part of the cell. The correct answer here will be C, endoplasmic reticulum. Alright, next, energy releasing reaction in a cell. Energy releasing reaction. Where is energy generated? In the mitochondria. Now I want you to tell me what was the name of the infoldings of the inner membrane? What did we call it as? Didn't we call those infoldings as cristae? And what did the cristae have on top of them? Cristae had numerous oxysomes. Or can we call those as F1 particles? F1 particles, or you may also call it as the elementary particles. Okay, you may also call it as the elementary particles. Alright, next, shall we go ahead? Now, next question. Ribosomes take part in protein synthesis in, tell me, ribosomes. First of all, these are cell organelles which have no membranes. Okay, so if they have no membranes, they are going to do the work of protein synthesis, but where are they found? These are structures, remember, they are found in both in prokaryotes as well as in the eukaryotic cells too. Okay, so ribosomes are found in both. They do exactly the same function also in both of the cells. Okay, prokaryotics as well as in the eukaryotes. Alright, next. Endoplasmic reticulum is always going to be remaining attached to, tell me, where did that endoplasmic reticulum start? Remember that part? The nuclear envelope. Yes, so that is where the endoplasmic reticulum is going to begin. So naturally it is going to stay attached to it. This is the endoplasmic reticulum and this is the nuclear envelope. Okay, so the correct option is this. Alright, next. Which structure performs the function? Which structure performs the function of mitochondria in a bacteria? Which structure performs the function of mitochondria? Tell me. First of all, mitochondria means that it is going to be doing the work of generation of ATP, right? Do you think that which of these you think actually is going to do that? The cell wall, the ribosomes, or the nucleoid? Do you think so that the nucleoid? No. The nucleoid just does the work of storing the genetic information. What about ribosomes? Ribosomes do the work of protein synthesis. What about the cell wall? That is actually going to be giving shape and structure to that cell. And so this also concludes that our last option would be the mesosomes. Yes, mesosomes are the region where the actual function of the mitochondria takes place, so that prokaryotic cell can be getting energy. Okay, shall we continue with the next? A cell organelle which is having or containing hydrolytic enzymes is cell organelle containing hydrolytic enzyme. Now remember one thing, these cell organelles which is having the enzyme is none other than the lysosome, right? Okay, these lysosomes, the lysosome, were also called as the suicidal bag, suicidal bags. Okay, and also you need to remember that it performs two types of destructions. So obviously lysosomes are meant for breaking down, for destruction. So there is one destruction which is going to be happening inside the cell. So you may call that process as the intracellular digestion. Okay, and there is another one which was called as the extracellular digestion. Okay, now when I talk about the extracellular digestion, what does that mean? You all are acquainted with the intracellular. This was in the case of the process during fertilization. Okay, so basically what happened is during fertilization, we see that the ovum, okay, the human ovum has got a membrane around it. Okay, so basically many sperms are going to be approaching this ovum, aren't they? So when we see many sperms are approaching the ovum, the sperms will release an enzyme, and that enzyme will be able to break down that membrane. So whichever sperm releases the enzyme, what is that enzyme called as? It is called as hyaluronidase. Remember, ASE means it is an enzyme. So the sperms are going to be releasing an enzyme called as hyaluronidase. That hyaluronidase has the ability to break down this membrane here, because that membrane around the ovum contains an acid called as hyaluronic acid. Okay, so hyaluronic acid is going to be broken down with the help of hyaluronidase. Did you understand this? Who is secreting hyaluronidase? It is the sperm. And what is it going to break down? The layer of hyaluronic acid around the ovum. Okay, now this is a perfect example where lysosomes are going to be in the sperm, which are releasing an enzyme which is going to not work on the sperm but work on a part in the ovum and around the ovum. And the sperm who is able to release that enzyme is the sperm which is going to be allowed entry inside that ovum, and all the other sperms will be kept away. So if at all this is the sperm which is going to release that enzyme, okay, let's just highlight it. If this is the sperm which is releasing the enzyme, then only this sperm will allow to be going inside that egg. All right. So that is how extracellular digestion occurs in the lysosomes. All right. Okay. Next, plasmodesmata connections help in. What do they help in? Plasmodesmata, these were those connections which were present between the cell walls in the plant cells. Do you remember that? Yes. What do they help in? They are basically present so that any substance can be carried from one cell to another cell. So basically it behaves as a bridge. So this is going to help in movement of substances which are there between two cells. Adjacent cells, whenever you want any substance to travel from one to another, then plasmodesmata is the one that's going to be helping in that. Okay. Next, Golgi complex participates in. What is the main function of Golgi complex? Do you remember that Golgi complex arised from the endoplasmic reticulum? Ahead it is going to be forming vesicles, I had told you. Remember, there are small shaped vesicles which are formed a part of the Golgi bodies. So here the correct answer, it is participating in the formation of secretory vesicles. Now remember one thing, the function of Golgi complex. It is the work of creating or generating complex carbohydrate from simple sugars. Okay, complex carbohydrates from simple sugars. Alright, next question. A prokaryotic cell lacks. What is it from these four things that a prokaryotic cell absolutely does not have? Let's see the options. True nucleus, does it have it? No, it doesn't. A nuclear membrane, well, there is no nucleus. How do you expect there to be a nuclear membrane? Membrane bound cell organelles absolutely are not present. What is present? Ribosome is present, and remember one thing, ribosomes do not have membranes around them. So the answer correct here would be all of the above. All of these cell organelles, or rather all of these options which are given are not present in the prokaryotic cell. Okay, alright. So children, this was the whole conclusion from the topic of cell, its structure and functions. We have gone through several MCQs which are going to be very useful for your MCQ, your exams point of view. And also what you can do is go to our YouTube channel or go to our Yupp Master app, where you have a collective of all of these videos of cell, structure and function. You can go ahead, you can watch the videos again if you require. You can also take screenshots so that you can write your own notes, make your own notes for this. Alright. Now with our next lecture will be coming soon, and in the next week we'll be starting a brand new chapter. So do stay tuned with me, and we'll crack this, and you guys stay home, you stay safe and take care until the next time we meet. Bye bye and